All right. So most of the lecture will be just me talking at the screen like this. Sometimes I will flip my tablet around and write on the screen itself. But our first topic is just talking about what DNA is in general. So DNA is the building block of all life. It forms what we call our genetic code, and it essentially has all of the instructions for how we appear, most of our behaviors, any adaptations, um, feedback responses, all encoded within these molecules. The actual DNA as a word is actually an acronym for deoxyribonucleic acid, and that basically means that it consists of nucleotides based on deoxyribose sugar. So you can see here, this is a strand of DNA that is sort of like a section of a strand of DNA. And you end up with three particular components per nucleotide. So nucleotide is like the base form of DNA. I'm just going to flip over my screen so I can annotate on the slides now. So these three components right here make up one nucleotide, or one base component of DNA. We start off at the very top with something called a phosphate group. Connected to a sugar with 5 carbons in it. So this is our deoxyribose sugar. Connected to this sugar as well is our nitrogenous base. So that is one nucleotide. And the bonds between the phosphate group and the sugar are covalent bonds, which are very, very strong. And the bond between each of these, oh, sorry, never mind. There are 4 kinds of nitrogenous bases. We've got adenine, thymine, guanine, and cytosine. And adenine and thymine will always bind together. Guanine and cytosine will always bind together. Between the nitrogenous bases, connecting 2 strands of DNA together, we've got hydrogen bonds. And while hydrogen bonds are still quite strong, they can be easily broken by certain enzymes, which is going to be a pretty important component later on. DNA is a double-stranded molecule, meaning that it's got 2 parallel strands connected together, and they start to spiral together in a shape that we call a double helix, which is this structure that you see on the left-hand side there. In terms of the structure of DNA, it is all based around this thing called a pentose sugar. So pentose sugars are ones with 5 different types of carbon in them, all in one carbon chain. So we've got ribose sugar, which is the base form of RNA, which is another type of ribonucleic acid. We'll talk a little bit more about RNA later on, but deoxyribose is the sugar found in DNA. We've got carbon number 1 on the far most right-hand side there, carbon number 2, 3, 4, and 5. And then to this carbon chain, we've got 2 things that are being bonded to. And if you look on this side right here, you can see the phosphate group on the top will always be bonding to carbon number 5. So it'll kind of be coming... Oh, there we go. It'll kind of be coming off the side of carbon number 5, and then the other group, phosphate group, will be connected onto carbon number 3, and then it'll continue the chain on. So basically, we orient or talk about the structure of DNA in terms of its 5' end, as in the phosphate connected to the 5th carbon, and the 3' end, as in the phosphate connected to the 3rd carbon. The interesting thing, though, the parallel DNA strands that are bonded together by hydrogen bonds are actually anti-parallel, or flipped over, essentially. So the 5' end of the strand on the left-hand side will only bond to the 3' end on the strand on the right-hand side. And as you go down, the 3' end on the left-hand side will bond to the 5' end on the right-hand side. So they're parallel to each other, but they're running in opposite directions, kind of like that. And that's important to know for your DNA replication section. So in terms of how DNA is stored within the cell, this is its structure in eukaryotic cells. So I'm just gonna flip over again so I can write. Okay. So we end up with DNA as a double helix singular molecule here, and the strands of DNA start to wrap around and coil around these histone proteins, these circular ones right here. They join the histone proteins together kind of by loop around them like so, until you end up with a very long chain of chromatin. As soon as a DNA strand will wrap around a histone protein, it is called a nucleosome when they're wrapped around together like this. Then chromatin will start to condense, so they'll start getting longer into these chromatin loops and squish together essentially, so they become more compact. And once chromatin becomes compact, it turns into a chromosome. Chromosomes have a couple of notable features. First is this center right here that connects the two chromosomes together. This is called a centromere. Um, here. And then finally, at the caps of the chromosomes, we have these structures called telomeres. Telomeres are caps that stop the DNA from unwinding back into chromatin, and centromeres are like sections of DNA that hold the two chromosomes together but don't actually code for anything. So this is our number one way that DNA is found in a cell, wrapped around histone proteins and condensed chromosomes. Histone proteins and chromosomes. Then we have our second way that DNA is found in the form of prokaryotic cells. So in the previous one, we were talking about eukaryotic or simple cells, sorry, more complex cells. These are our prokaryotes, so bacteria and things like that. There are actually two ways that DNA is found. Both of them are in the cytosol of the cell if you want location. The first one is in the form of plasmids, which are these circular rings of DNA. And second way is unbound DNA found in the chromosome. Unbound DNA found in chromosome. So that's our second way. The QCEE actually has three different ways that DNA can occur, and this was a previous exam question. The first one, as we discussed previously, is in eukaryotic cells, wrapped around histone proteins. Histone protein in chromosomes. The second one is actually in the mitochondria. So every single eukaryotic cell will have mitochondria, which undergoes cellular respiration. They kind of look like this. And all mitochondrial DNA comes from your mother. So it's descended down the family without any influence from the sperm cell or from the father's genetics. This basically means that your mitochondrial DNA will be identical to your mother's, identical to your grandmother's, and this can actually be used to test for evolution or relatedness between organisms. So mitochondrial DNA, which is found unbound in organelles. The third way is as whoopsie. plasmids, and unbound DNA in the prokaryotes. I strongly recommend that you note all of these three together. They came as a 2021 um, external paper question. And it was literally worded in the way that it was like, how does DNA occur in the cell? Which basically meant name the three ways that DNA is represented in the cell. And it got a couple people confused, so watch out.